Hello everyone, can you hear me well?
Yes.
Okay, so to start my presentation, I would first like everyone who knows or have heard about CRISPR or CRISPR-Cas9 to raise your hands. Okay, there are some of you, but it's not the majority. Before the summer of 2012, I'm sure that nobody here would raise your hands. This is because in July 2012, there was an article published with a very exciting content and not so such an exciting name. But this article published whose author was Emmanuelle Charpentier and Jennifer Doudna, changed the world of biological sciences. They introduced a concept that was to change the world of gene editing, gene therapies. People were hoping for breakthroughs in cancer therapies, in agriculture, in scientific research, in biological research, but not all of this happened. Actually, when it comes to medicine, the use of CRISPR in medicine, the first large breakthrough happened on December the 8th, 2023. So over 10 years after the initial discovery. So you can see that, for example, in the case of artificial intelligence, the breakthroughs are happening very, very quickly. So, for example, ChatGPT, it has evolved so much in less than a year. But with biological sciences, with gene editing, and with CRISPR, it's not that rapid. We have to wait for breakthroughs. Of course, CRISPR has already changed the world of agriculture, of scientific research, of preclinical trials, but it hasn't changed the world of medicine yet. And today, I would like to explain to you what CRISPR is, to those of you who do not know what CRISPR is, and then discuss why, in fact, we have to wait so long for scientific discoveries, for medical therapies that use CRISPR. So, let me start by saying what CRISPR is. So, here I'll have to introduce a few facts about human DNA, human genome. I'll use some simplifications to make it more clear to you. So, imagine that our DNA is created of bases. So there are 4 bases that create our DNA. This is adenine, thymine, guanine, and cytosine. So for simplification, just think of them as 4 molecules that create our DNA. And we have 3 billion base pairs in our genome. So we have 3 million pairs, overall 6 billion of these molecules in our genome. And we want to edit one of them. And how do we do it? Here's where CRISPR comes. It was first discovered as a defense mechanism in bacteria. So in the early 2000s, scientists have discovered that bacterial DNA often has these short repetitive sequences. And they weren't really sure what that is. But then Emmanuelle Charpentier and Jennifer Doudna, they took on that research and they continued it. They realized that actually the repetitive sequences are parts of viral DNA. So they have discovered that a bacteria was able to take some of the DNA of the virus that attacked it and incorporate it into its own DNA. So the bases, the molecules that once were in viral DNA, could be transferred to the bacterial DNA. It's a defense mechanism because when this bacteria is attacked again by a virus, it is easier for it to trigger a defensive reaction. And it has been obviously a breakthrough in the world of microbiology. But Emmanuelle Charpentier and Jennifer Doudna took it on another level. They have noticed that this method of cutting DNA, of incorporating one part of DNA into another strand of DNA, could be something that we can use in the world of biological sciences and in the world of gene editing. So let me now explain how CRISPR works and how it can be used to edit our DNA. So we have the CRISPR part. The CRISPR part is a strand of RNA. It's pretty similar to DNA. It just has the different, instead of thymine, it has uracil. But it's pretty much a strand, one strand of molecules that guides, that shows you which place is going to be cut. So it is very important to know which of this, in which place among these 3 billion pairs of bases, 3 billion pairs of molecules, the Cas9, which we'll discuss in a second, will have to cut the DNA. So CRISPR is the guide. It shows you where the cut will be made, where exactly in our DNA, in this long strand. Then we have the Cas9, the second part of the name. So the Cas9 is a protein, and it's something that's able to cut DNA. So once we have CRISPR and we know where to cut, we then introduce Cas9 to cut the DNA in the place we want it to be cut. But there's one more thing that counts here. So when the cut happens, the DNA has the ability to come back to its shape. But if we introduce a new base, a new molecule that we want to incorporate in the place where the cut has been made, it automatically can be incorporated into the DNA strand. So basically what I am talking about, what we can do here, is that we can change that one molecule in our DNA, one of these bases, into another one. And why it's so important? Because there are diseases such as sickle cell anemia, such as cystic fibrosis, that are dependent on just one base. Out of our 6 billion bases in our DNA, just one contributes to a disease that alters your whole life. So there have been hopes back in 2012 that we might address these diseases with CRISPR. And of course, it is true that the road from the lab to the patient is extremely long. It can take decades. And as we can see, for the first therapy to be approved by FDA, so the Food and Drug Administration in the US, it took more than a decade. And now I will show you what it takes for a drug to be transferred from the lab, from the initial idea, from the CRISPR-Cas9 that was discovered in 2012 to a patient. So the first step that we have is the discovery and development. So once scientists have an idea for a drug, for a therapy, they know how CRISPR works, they need to now use it in the lab and try to think of the targets, of the proteins that it may target, of the places that can be changed, what diseases it can be applied for treating. So this is the first part that happens in the lab. Scientists test their initial hypotheses. They try to think of possible places, possible bases they can change, possible diseases they may cure. And once that part is complete, once they have a clear idea of what disease can be treated with CRISPR-Cas9 and which base or which part of DNA needs to be altered to do that, they go on into the preclinical research. Of course, I'm describing it very shortly here, but each of the stages can take several years, even up to a decade. Then we have preclinical trials. So this is the part where the drug that we have, the therapy, the idea that we had, gets tested. It's tested in cell cultures, but it's also tested in animal models before it can be tested on humans. So it's a very long part of the process, and it also takes several years usually until we can get to the clinical research, so clinical trials. And even within the clinical research, we still have 3 phases that each therapy needs to go through. So first we test it on healthy individuals, Then we tested on people who have the disease but on a very small group, looking for side effects, looking for potential problems, if it's effective. And did you know that up to 80% of drugs of therapies actually gets rejected at this phase? So everything that has been done before just isn't proven effective, and scientists need to start over with a new therapy. But fortunately, the new CRISPR-Cas9 drug has passed also the clinical research phase, and it has been granted the FDA approval. So it has been approved for use in patients who are suffering from sickle cell anemia and beta thalassemia. These are two gene-related inherited blood disorders that, for example, can cause not enough of beta globin to be produced. This is a protein in your blood that is responsible. That is a part of hemoglobin, which is responsible for carrying oxygen in your blood. So basically, when you have these two problems, you can be treated with the new drug. But even when the drug is approved, it's not the final stage. We still have the post-market monitoring. So once this drug has been approved and has been introduced to the market, the FDA still continues to do research, to test, to look at the effects, to look at possible side effects. So overall, as you can see, it may take up to several decades until we get from the idea in the lab to the actual drug and actual therapy. So there are advantages of such approach, because with gene editing, there comes a lot of ethical problems. So we have problems related to editing genome as such. For example, in 2018, there was a scientist in China who claimed that he had produced two twins who were genetically altered. So he altered the children before even he altered the cells from which the children grew later. And he got imprisoned for that, and he lost his license, and he's unable to do research right now and to perform further research on this topic. That's because the topic of gene editing is so subtle, and it has to be approached very with a lot of delicacy. People are against it usually. There are a lot of problems also related to risks of cancer. So for example, where there are in previous gene therapies, not the ones using CRISPR, but gene therapies that use different gene editing techniques, there were cases where patients developed tumors afterwards or cancers. So this is what we still fear and what might come. But having a lot of time, a lot of decades, we can prepare for these changes, and we can make sure that every therapy is introduced ethically and that we don't have legal or ethical problems with it once it's introduced. This is, for example, a problem with artificial intelligence. It's developing so quickly that we can't control it, we can't regulate it. With gene editing, we have more time to do that, more time to think about what are the ethical and legal problems. And even when we have the drug, there is one last problem that comes into the place, that every therapy, every gene therapy is extremely expensive. The new drug, Casgevy, it's estimated to cost $2.2 million per one patient. And of course, a patient is not able to pay that on their own. So if, for example, the government won't be able to sponsor, to fund this treatment for the patients, such a therapy may be simply cancelled, may not be used anymore because it's not economically beneficial for the company that created it. So there are a lot of things that may go wrong when creating a gene therapy based on CRISPR-Cas9 or any other type of drug, to be honest. So we need to be prepared, we need to be patient, and we need to wait until every therapy goes through all of these steps. And I believe that we should all hope for changes, although they might not be there yet, but there are a lot of clinical trials that test CRISPR-Cas9 therapies for example, lymphomas, for leukemias, so it can also address cancer, for many other rare diseases such as cystic fibrosis. And although there are a lot of steps, there are decades, there are years that need to take before we go from CRISPR to an actual therapy, I think it's a direction in which the science will be going in the following years. So what I want you to take from my talk and what I want you to do maybe today in the afternoon, it's just to read a little bit about CRISPR, because arguably it's the most important discovery in the biological sciences. It was awarded the Nobel Prize in Chemistry in 2020. So it is going to change the world of biological sciences. It has already done so, and I'm hoping that in the following years it will change the world of medicine and the fate of patients with cancers and with rare diseases. And I think we should all take a moment to just read more about CRISPR and see what's coming in the next years, in the next decades. Thank you very much.